Welcome everyone to the CIBM Breakfast Science Seminar Series Summer Edition, which will be featuring PhD students affiliated to the founding member institutions. The second speaker today is Hawley Holman from the Image Guided Interventions Laboratory at the Department of Radiology and Medical Informatics at HIFU. His PhD supervisor is Professor Raris Salomir, who was the first speaker of the 2021 Breakfast and Science Seminar Series. Thank you, Ryan. The floor is yours.
Hi, my name is Ryan Holman, and I'm a member of the Image Guided Interventions Laboratory at the University of Geneva. This presentation is titled Improving HIFU with a New Perfluorocarbon Adjuvant, and it describes experiments evaluating perfluorocarbon emulsions with a unique type of fluorinated surfactant for focused ultrasound therapies. So I need to acknowledge my funding agency and also mention that this presentation is part of an outreach activity as part of my fellowship program. And the funding agency for my position is provided by the UCOM Consortium, which is a Marie Sklodowska-Curie training network aimed at doctoral training for studies related to ultrasound cavitation and soft materials. The UCOM Consortium is part of the EU Horizon 2020 Research and Innovation Program. The hyperlink for the consortium website is found here. And also I've made a GitHub code repository for some of the software that I've developed on this project. These droplets are designed to be administered intravenously, and they are thought to be advantageous, particularly for treatment locations such as intra-abdominal tumor sites, which can be complicated by the need for high power requirements and need to account for respiratory motion and account for interaction with the rib cage. So the concept is that these droplets would be applied to remain in systemic circulation and allow a lower power requirement from the focused ultrasound transducer, or reduce the treatment times necessary to perform the ablation. So these droplets feature a perfluorocarbon core shown in this molecule, and then the surfactant that are used is this F-TAC molecule, and it features these fluorinated tail groups that interact with the perfluorocarbon, and then the hydroxy groups which stabilize the surfactant in aqueous solution. And the size of the droplets can be adjusted by modifying the repeating TAC units here. And so this is an example of a particle size distribution measurement performed with in a previous study with these droplets. So this particular example had about 2 to 3 micron mean diameter, and most of the droplets were between about 1 and 10 microns in size. And so these droplets have been studied, these type of droplets have been studied for quite a while for blood tissue oxygenation. And it's been shown that these emulsions can deliver high amounts of oxygen to hypoxic tissue sites, and these applications were studied for many years in order to evaluate the toxicity of these droplets, and were tested in clinical trials, and some forms are actually even approved by the FDA as a blood substitute. And more recently, the perfluorocarbon has been approved, has been granted orphan designation by the EMA for respiratory distress syndrome and congenital pulmonary hyperplasia. And so the clearance route and half-life of this perfluorooctylbromide molecule, which is the main component of the emulsions that we use in our study, has been shown to be very non-toxic and has a very short half-life. And it's reasonable to assume that the toxicity profile of our particular formula would be quite similar to some of the previous reports with similar emulsions. So last year, our group published a paper in the International Journal of Hypothermia describing improved heating during magnetic resonance high-intensity focused ultrasound. after administering these perfluorocarbon emulsions to the perfusion fluid. The emulsions were pumped through the ex vivo pig liver and ex vivo pig kidney, and the temperature increase was compared to the control cases and also to a perfused tissue mimicking phantom. This figure illustrates some of the main findings from this article. Top row compares the temperature increase due to the presence of emulsions in our perfused tissue mimicking phantom. The middle row compares the temperature increase due to the presence of emulsions in ex vivo pig liver. And then the bottom row compares the temperature increase due to the presence of emulsions in ex vivo pig kidney. And in the ex vivo organs, we saw an increase in thermal enhancement by about a factor of 3 compared to the control case. And the amount of droplets administered were about 0.2 volume percent concentration in these applications, and it's very similar to doses administered with lecithin PFOB emulsions during blood oxygenation studies. This is a good video showing the boiling core effect occurring while emulsions are circulating in the kidney vasculature. This signal hypointensity occurs due to susceptibility artifacts that are generated as a result of fluid vaporization at the Haifu focal spot. This is an ultrasonography video of focused ultrasound sonications in our tissue mimicking phantom. The sonication pattern is circular, and the focal spot can be seen moving in the circular pattern. And so the signal change remains even after focused ultrasound sonication ends. And one of the main objectives to the study is to determine if this vaporization that occurs is probably resulting in this signal change. If this vaporization is occurring within the droplets, vaporization of the perfluorocarbon fluid within the droplets, or if it's due to vaporization of the fluid surrounding the droplets on the exterior of the droplets. In the next few slides, I'll describe some of the main results from a recent research article that's currently under peer review in the International Journal of Hyperthermia. And in this article, we were able to arrive at a dose response model for in order to match the thermal enhancement at varying concentration of emulsions in the perfusate of our tissue mimicking phantom. And also we were able to get a measure of the size of this interaction volume that we've observed in previous studies, and compare the size of this enhanced heating volume to the actual radius of the microspheres. So this image is basically just describing the experimental apparatus that we used in order to evaluate these experiments. And so it's the top image is a 3D volumetric rendering of our perfused tissue mimicking phantom, which I described briefly in some previous slides. But the bottom two images are the cross-section of that phantom, and the bottom left image shows the cross-section of the phantom in the presence of air bubbles in our perfusate fluid. And then the bottom right image shows a comparison of the cross-section of our phantom after the air bubbles have been removed from the perfusate. So the presence of these air bubbles are pretty problematic for magnetic resonance thermometry. They can render the temperature measurements invalid and cause a lot of MRI artifacts. And so we have to go through great lengths in order to ensure that these air bubbles are not present prior to actually performing our measurements. This figure illustrates the change in acoustic absorption coefficient as a function of emulsion concentration in the perfusate of the tissue mimicking phantom. So this absorption coefficient is based upon the temperature increase in the region surrounding the focal spot, but it's also a function of the HIFU applied power and HIFU duty cycle. So it allows us to get a comparison of what sort of absorption and temperature increase we might expect at varying concentrations, and also at sonication conditions for different experiments. And so it's one main important aspect that we observed from this illustration is that the droplets tended towards an asymptotic value at concentrations not much higher than we used in our experiments, and that are also quite a bit less than values used in experiments with less than perfluorocarbon emulsions for blood oxygenation. And another important finding that we obtained from this illustration is that we're able to generate a continuous function of absorption coefficients that we might expect at all of the emulsion concentrations within our range of interest. So this table illustrates some of the sonication conditions corresponding to the curves that were shown in the previous figure. So the previous figure had a set of conditions 1, 2, and 3, and these were the Haifu sonication conditions that were used to obtain those data points. And also shown are these fit constants that were derived from the mono-exponential function that I discussed in the previous slide. And lastly, we did a approximate measure of the size of this interaction radius, or it's equivalent to the radius of the temperature increase, the volumetric temperature increase surrounding each individual microsphere, and how this this relationship changes with these fit constants and actual droplet size. And again, it's not very intuitive just given the information that's presented here, but it's described more in the publication. But the primary basis was not to get a very accurate measurement in this instance, but it was primarily just to get a ballpark figure so that we could know what size interaction volume we might expect when we perform our simulations to match our experimental data. This illustration illustrates the algorithm that we used in our simulation in order to arrive at the interaction radius size. And basically the components of the simulation that were adjusted were the size of the interaction volume and also the concentration of droplets. And then we normalized all the data and then compared it to the experimental findings to see which set of interaction radii best fit the experimental data. And when we did all the simulations, the figure in the bottom left here with the blue figure is a single slice through one of these three-dimensional matrices, and each of these yellow points represents one microsphere. It doesn't represent the actual microsphere, it represents the probability of temperature increase in the vicinity of the microsphere. So it was given a three-dimensional Gaussian probability distribution centered at the central coordinate of each of these microspheres, and then each of the microspheres corresponded to a specific concentration within this matrix volume. And then we summated all the pixels in these volumes at each droplet concentration and each interaction radius, and and then overlaid them all in the same figure and then compared them to the experimental data. And that's illustrated in the next figure. This figure is the results of our Gaussian kernel simulation, and it's compared to the experimental data that I discussed briefly previously. And so again, we adjusted the size of this interaction radii, which is the radius of the heating volume surrounding 1 microsphere, and we adjusted it at varying concentrations of droplets in our simulation matrix. And then all the data for the simulations were normalized to the maximum value, and then the experimental data was overlaid, which this value eventually reaches an asymptote of 1, where there's no additional heating at some point at higher concentrations. And so we noticed that the trend was very similar to the size interaction radius, and this radius was also well within the range that we expected based on previous experimental studies, and also based on just a simple analytical formula that we derived in our article. And so this is an empirical approach of how the interaction radius was estimated based on this experimental data. In the next few slides, I will describe some preliminary results using these perfluorocarbon emulsions in conjunction with magnetic resonance acoustic radiation force imaging. So in order to obtain the displacement and temperature measurements using acoustic radiation force impulse imaging with these perfluorocarbon droplets, we had to write a custom MRI sequence, and this is a sequence diagram that we used, and it was based on a standard flash sequence, but we incorporated these motion encoding gradients into the flash sequence. And we performed scans during focused ultrasound sonications and post-processed the data, and the phase shift that results from the focused ultrasound can be equated to the displacement and temperature measurements using these formulas. And so we applied these formulas in a post-processing MATLAB script in order to analyze the data after the experiments to get the measures of displacement and temperature from acoustic radiation force imaging. And we compared it to standard magnetic resonance thermometry sequence in order to verify that the gels had the expected response from the focus ultrasound. But in addition to actually making this MRI sequence, we also had to make a custom gel formula that could be used for both acoustic radiation force impulse imaging and also for standard magnetic resonance thermometry. And it also had to be stored for quite a while so that we could reuse it for a few experiments in subsequent weeks. And so the displacement and temperature properties for the RFE sequence for the gel to be used with the RFE sequence were sort of initially based on measurements we obtained in ex vivo pig myocardium and pig kidneys. And so there's an example of some measurements that we performed in ex vivo pig kidney, where we measured the displacement and temperature, simultaneous temperature measurements for this particular sample. And then we made this tissue mimicking phantom. It's kind of distinct from the ones that I mentioned previously, but it's also not very different. It's a gelatin-based hydrogel, and it contains condensed milk and starch. Those are the main ingredients, but it's actually powdered milk, not condensed milk. The powdered milk in the gel itself is quite susceptible to bacterial infection, and so it's quite difficult to maintain the gels for quite a long time without using rather powerful powerful preservative. So we're still actually kind of formulating the recipe, but we were able to get some of these preliminary results that were sort of similar to what we hypothesized might be the response. And so this image on the top is the phase images resulting from standard magnetic resonance thermometry, which is just it can be equated to the temperature increase at the focal spot in the coronal plane and the transverse plane during Haifu sonication. And then these images down here are acoustic radiation force impulse imaging displacement and temperature maps in our phantom. So this is kind of the images that we generate and in order to analyze the data. So after we developed our MRI sequence and our tissue mimicking gel formula, we were able to get some exploitable results with our experiments. And what we did was we embedded these perfluorocarbon droplets in the gels at increasing concentrations, and then we performed magnetic resonance acoustic radiation force imaging and also standard magnetic resonance thermometry on these gels at all these concentrations. And we performed multiple sonications at each concentration to see how the results changed at the same gels and at the same focal region with multiple sonications. So this is just the standard displacement, and we kind of saw that it sort of increased on average with increasing concentration, what we might expect, and but also with each sonication, we kind of saw a downwards trend with succeeding sonications. And then this is just a measure of the integral displacement. So it's like this is only focal displacement. It's the maximum displacement at the Haifu focal spot, and then this is summation of all the displacements in a region near the focal spot. And then, like I said, this sequence allows simultaneous temperature measurements. So this is the heating in the gel with increasing concentration, and also with each sonication and at the same location in the same gel. And the temperature is a bit more difficult to get accurate because the heating, it does not heat very much, and I think there's kind of a lower limit on the amount of temperature increase that can be measured in this particular instance. But at higher concentrations, it's not as it doesn't affect the data quite as much. And so this is similar. This was the focal spot maximum temperature, and this was the heating in the region surrounding the focal spot. And so this figure is the four figures shown on the previous slide, but it's the averages at each concentration. So I averaged each concentration and then normalized all the data to their maximum so that I could put all the data points on a single plot, and then the tables down here correspond to the actual absolute values obtained at each of these data points. So these are averages at each concentration, and then this figure shows the standard magnetic resonance thermometry performed on the same gels with varying concentrations, and it was essentially used in order to verify that the temperature showed an increasing response like we've seen previously with higher droplet concentrations, so that that we were getting a proper response from the gels. Fluorine isotope MRI is a useful technique for verifying droplet concentrations in the focal region. The emulsions are composed largely of fluorine atoms, and in these images we compared a control sample to a perfused phantom with an equivalent amount of emulsions in the interstitial space of the phantom. The signal was near the expected values, and the primary limitation was shown to be the long scan times required to obtain these images. Another useful application of this technique is to evaluate remaining fluorine signal post-treatment. I think it's beneficial to discuss how the substance might be translated into the clinic. So a few clinical studies have been performed using perfluorocarbon adjuvants focused ultrasound in patients. The primary reported benefit seems to be time reduction in procedures like uterine fibroid ablation, and administration of our droplets in human volunteers would rule out adverse side effects. A feasibility study in a single patient would be the first application of perfluorooctobromide microdroplets as a focused ultrasound adjuvant.